Everyone. Welcome to the seminar. It's my pleasure to introduce Dr. Dr.Twarock, who's here from the University of York, where she's a professor of mathematical virology. She's gotten her PhD in mathematical physics, and she's worked a lot on applying mathematics to study viral geometry when it comes to the geometry of the virus particle, but also other things relating to viral genomes, for example. She's had multiple research fellowships, including the Royal Society Research Fellowships. And I'm hoping today we'll hear more about her talk relating to, again, geometry of viral particles, as well as the role of viral genomes, specifically structurally, as well as using packaging signals and other things like that in the viral life cycle. So please join me in welcoming Dr. Twarock.
Thanks. Thank you so much. It's a great pleasure to be here. I've been at a MUS conference the last couple of days, and I thought it would be wonderful to stop by and see you to talk about the projects we are doing in integrative systems virology, I would say. So summarizing what Tara just said, for me, is bringing together these different techniques from mathematics, computational physics, biology to answer questions in virology. So hence the title Integrative Systems Virology. And what we are wanting to look at specifically today are the multiple roles the genomes can play in viral life cycles. It's a longer story for us. Should be on. Yep. So what we are after is mechanistic understanding of viruses and life cycles, and we're in particular focusing on RNA viruses because in this case the RNA is packaged at the same time as the particles assemble. So there are very interesting interactions between the protein layer and the RNA that we want to discuss here. And our main goal is, and I hope you see this in this talk, to find novel drug targets that are conserved across strain variants so that we can potentially get broad spectrum antivirals. Let's focus first on assembly. And when we started in this field 20 years ago, I should say it's a story of 20 years, the view was mainly protein-centric. People were thinking of virus assembly as protein building blocks coming together to form these containers. And the question then is, how does a virus do this efficiently and swiftly? And the first system that showed us that there is more to it than just the bringing together of protein building blocks is actually a phage. And I want to start deliberately with this phage because it tells us about mechanisms that we later on discovered in human viruses, and I'll talk you through this story. But it's very instructive to see the phage first because you can see mechanisms that you would otherwise not see because they're much more complex in these human viruses, but it's a guide then what to look out for. So the hint you get here is a phage that assembles from a protein dimer that occurs in two types of conformations. You have 60 copies, oh, it's not working very well on the screen, 60 copies of the asymmetric form, that's around the 5-foot axis, and we have 30 copies of the symmetric form. Both interconvert in this assembly process. If you have protein in isolation in a test tube, it takes of the hour of days to see particles. But if you have the genetic material present, in particular certain RNA secondary structure elements, you only need to wait a couple of hours to see full assembly. So clearly, the genome has an impact on the efficiency of assembly in this case. And we started off trying to understand how this works. Now, in this particular system, what we need to know is how this interconversion between the symmetric and the asymmetric dimer occurs. So basically, if we look at the normal modes, that's what we've done in our team, that we look at these FG loops that you see on the sides here. So in a symmetric dimer, you have symmetric or antisymmetric motions, symmetric and antisymmetric motions. But the moment a packaging signal binds, an RNA stem loop binds, it asymmetrizes the motion. So the kinetic energy is displaced. One of the arms explores a larger conformational space, the other one a smaller one. And the one that explores the larger space has a higher probability of doing this flip and locking in this assault bridge. So basically it promotes this switch from the symmetric to the asymmetric dimer. Now, with this in mind, then, we were asking the question, what if that happens multiple times across the genome? So here is an early cryo-EM reconstruction from the Leeds team, Neil Ranson and Peter Stockley. You see the protein layer on the outside, the RNA on the inside. You see these contacts here. And basically, if you look at the contact sites between RNA and protein and connect nearest neighbors, you get a polyhedral shell. And my team then developed a new data analysis algorithm that uses geometric information from that polyhedron to interrogate the genome for potential cryptic signals, for the location of potential packaging sites. It is very much a problem that's known as Hamiltonian path analysis on a polyhedron for the mathematician. Hamilton himself had this as a board game. So the viruses basically play the Hamiltonian path game that Hamilton envisaged at that time. So to give you a little bit more detail how this works, so what we have is from bioinformatics candidates, potential candidates for packaging sites. We know from biochemistry the rough determinants of what we expect from a packaging site, or we're looking at a known high affinity packaging site and see what characteristics it has. Then we look for other candidates, and typically you have way too many, you have more than double of what you actually expect, or it's not quite clear what the packaging sites are, if any. But with this technique, you can weed out a lot of options just on the basis of geometry. So geometry is like a lens, it's like a microscope for us to do, if you wish, geometry-informed bioinformatic analysis in this case. And by doing so, we've been able at that time to pinpoint the positions, you see them in the secondary structure of this phage down there, the positions of these potential packaging sites. And what's really interesting is that these sites are very cryptic. Sometimes they have only a Y and an A, so pyrimidine and an A in a specific position with regards to a secondary structure element. Very difficult to find by bioinformatics analysis alone. But if you bring in knowledge of viral geometry and of these kind of analysis techniques, you are actually able to tackle that problem. Now, we understand as well how this mechanism is working. So we have done computational modeling on the one hand, where we use Gillespie-type algorithms on a coarse-grained version of a virus. So imagine a dodecahedron assembling from 12 pentamers. And the genome is just represented as a string of beads, where every bead is encoding an affinity band, the probability of attachment to the protein. And if two proteins attach at the same place, the proteins can also bind to each other and nucleate, assemble, and start assembling. So these are reaction kinetics models that we are tackling with Gillespie-type algorithms. So what we've shown then in this work here is that the affinities are tuned like knobs on a radio. So they need the right affinities at the right positions in order to be efficient. It is not enough to be having a strong affinity all over the genome. That would not work. You would need a mix of strong, intermediate, and weak. We understand that this has to do with nucleation process, that has to do with overcoming kinetic barriers, overcoming dead ends in the assembly process. So if you're interested in that, talk to me. I'm around a little bit. I just want to cover more ground and come to the human viruses, because I know you want to see the human viruses and not the phages and these models. But if you're interested in that, please talk to me about this specifically. The other thing we've shown is that if those affinities are tuned just right, then it allows the virus to overcome competition from competitor RNAs. So if there are cellular RNAs that can mispackage, and by random chance have some of those motifs, they will be basically outcompeted if this distribution has evolved in the appropriate way. Then I would like to advertise experimental work by my colleague, Professor Stockley from Leeds, where they show yet another feature of these packaging sites. So they take the MS2 genome and they add either the cognate protein, the MS2 protein, or they add the protein of a plant virus, STNV, and they measure the hydrodynamic radius. And what's really interesting is you find a collapse of the genome, compaction of the genome, when you add the cognate protein, whereas you don't see this with the non-cognate protein. These features are generic, but when we are coming to human viruses in a minute, you will see that they often do those mechanisms in different ways. So SARS-CoV-2 would supersede this with N protein compacting the genome, so to speak. So a much longer genome, a much more complex system. So some of those functions then will be substituted perhaps by another protein unit. But in any case, it's all these sort of functions that the overall mechanism is delivering to you. Or if you've got viral factories, then sometimes you don't need the selective packaging. But again, all of it is delivered in one way or the other to you. So now, in a nutshell, what I've shown so far, and that's the end of my sort of introduction to this talk, is that what we discovered together with the Stockley lab here is that in the linear sequence of the RNA viruses are cryptic signals. They are often as cryptic as just two letters. In this case, it's an AXXA in this example. That if they are presented in the appropriate secondary structure elements, so in this case, they all would want to be in the loop of a stem loop, then they would collectively promote virus assembly in this case. Now, as you can already see from this example, the variation in structure is quite strong. So you see that the actual individual stem loops look rather different, but what they have in common is the position of those recognition sites with regards to the element. What I haven't got a slide on also work from the Stockley lab. They have added mononucleotides in between them. They've changed the distances, and that ablates assembly as well. So you need the exact distances, which probably you already guessed from what I said at the beginning. We use those distances actually as input to get those sites because we know the distances are really important. So with this in mind then, let us look what we've done.
Right.
Oh, yeah,
Good
yes,
question.
please.
Can you use that signal, that knowledge to actually build a complete synthetic system?
Oh, that's the end of the talk. Wait a minute. Yes, we can. And we can also, for gene therapy purposes, take therapeutic cargos and code.
Okay.
We have been up early this morning on one of our systems. We do work with gene therapists in London at Great Ormond Street Hospital. I can tell you a little bit later. And yes, it's possible. There's some data at the end of it. Yeah. So exactly, so that's where we want to go. So the problem is at this point, so in order to do what you just asked, Melanie, we need to know for every viral family what the sites are, because you need to know what to code in, and that's specific for a family. So it's not the same across. So this part of the talk now is how do we find the needles in a haystack for virus you might be interested in, and then basically we use that knowledge either for antiviral therapy or for recoding. Yeah. So these are viruses that we've been working on, and I've put them deliberately because I think maybe some of you work on some of those viruses, and there's probably more to discuss with different people here. So let's look a little bit at the techniques we've developed. So first we looked at picornaviruses. That's a family of viruses where structural data was available to us. So we see different picornaviruses here, and the cryo-embryo reconstruction clearly revealing contacts between the genome and the protein layer here. And in particular, there is density around the 5-fold axes. So the question is, are there repetitive contacts again, or is this a random motif here? So what we've done here, this is joint work with the Stockley lab, is the integrative part of the talk, where the Stockley lab uses RNA select. So we have randomized RNA sequences that are then offered to protein. Whatever binds is amplified in 12 rounds of amplification. So at the end, we get large data sets of RNA sequences that we know have bound to protein, but we don't know which parts have bound. So just again, looking for a straight bioinformatics analysis is difficult because it's cryptic motifs. So we've developed a pipeline here. We call it the Bernoulli analysis. So that's, in a nutshell, you look at matches that are non-contiguous with a certain equivalent probability equivalent to a certain contiguous match. So in particular, these are Bernoulli peaks of 12, which means it's a non-contiguous match, maybe across 80 nucleotides, with the probability of a 12 match in the sequence. So what you see then is, if you do this, you slide along the genome, you see the genome on the x-axis, and on the y-axis, you see build-up of these Bernoulli matches. So if there is a match of that type, we start to build up these peaks. And then we can go into these sequences, take out maybe 50 or 100 nucleotides around the peaks, and do what we call a similarity analysis. So this is taking into account conservation, different strains, so it works better the more strain variants you've got, and it takes into account what folds we're profiling the RNA secondary structures in these areas and look for commonalities. So with this in mind, we've been able actually to, we take the highest peaks first because these are where the motif is usually most readily visible, because there are not so many alternative folds that would give us complexity. So with this in mind, we got those types of structures there, which all have a consensus GXU recognition motif in the 5' portion of the loop in this case. And building this into the cryo-EM density, that's again work in leads, you see the molecular details of the fit here. So basically with this joint pipeline, we can actually characterize those contacts to molecular detail, to full detail here. Now, the interesting question, however, for us is then, how conserved is this? Is this then therefore something we have to redo on every different strain variant? Or do we actually have a common theme here? So HPV3, Sarah Butcher lab worked on that. So we saw these contacts there. And what we then did here is we looked at the proteins in that family, and we did basically a distance graph on how different the amino acid sequences of the capsid proteins are. That gives you this graph in the middle, and you see that from this point of view, HPV3 is most distal from the case that we've just discussed on the previous slide. So we're going to the case with the most variation, if you wish. So what we then do, we take the HPV3 genome, we take the selected library from the experiment for HPV1, and build up these Bernoulli graphs again. And what we see when we then look at the highest peaks is again the same recognition motif, the same signal. So we can actually show that there is conservation of that motif across different strain variants. And that's why we are so interested, because that's a potential to get at broad spectrum therapy. Oops. Now, what's really interesting about picornaviruses, so we're starting already to get at viruses that deviate from this phage mechanism that we've seen, is that there are two types of such signals. So there is the ones around the 5-fold axis, but actually there is another location around the 2-fold axis where we have another signal. So we did a similar type of analysis, but what's really, really exciting here is that what is binding is a single purine in the loop. It has to be exposed in a specific way in the tertiary structure. So this is very exciting for the theoretician, because we are working on new pipelines to develop an analysis on 3D structures of these packaging sites here. And actually a new PhD student with Excientia is now working with me on an AI-based approach to identify in these cases in a high throughput way, those packaging sites. But what's very interesting again, so if we go into other picornaviruses, so we take rhinoviruses, foot and mouth virus, by the way, we have a joint grant with Toby Tuttle from the Purbright Institute on that, or other picornaviruses, then we see conservation across all picornaviruses of this size. So that's very exciting again for us, because poliovirus and all these type of viruses are amenable to the same analysis. We find both type of signals in all these different viruses. In particular, also we have this Roscoe Award for rhinoviruses to basically look into details there. Now, what I've said so far then is that we have different variants of a mechanism. So we went away from just having a phage mechanism to looking at different ways this mechanism might work. It differs both from the point of view of the host. So we have a phage, plant viruses, human pathogen. We saw one example until now. But also the way the mechanism works depends, and that's where my expertise on geometry comes in, works with the geometry of the building block. So the MS2 works with dimers. So it's a protein dimer mechanism with a kinetic sort of switch between two types of dimers. There are also situations where, and that's very common in other viruses as well, where we have monomers that come together as trimers, where the trimer formation is promoted by a packaging site. And then finally, we've seen in more detail the pentamer formation. So we have different types of signals, different types of ways the mechanism is implemented for different geometries here. You already see a certain level of variation. But then the question is, what happens with DNA viruses? So hepatitis B viruses pack pregenomic RNA. So it is, from the packaging and assembly point of view, behaving like an RNA virus. But it's classed as a DNA virus because it reverse transcribes within the particle. So, but because on the packaging level it packages RNA, we've asked the question, does this virus have that same mechanism as well? So we have again carried out the selects analysis, so the pipeline I've described before with the similarity analysis, and we found this RGAC motif. I should say it's an RGAC motif. So then we looked at the positions of those signals, and what's very interesting, they are more sparse. They're not as many. And the reason is that it reverse transcribes. It doesn't want to glue itself too much on the inside of the capsid. It has to be able to do this reverse transcription mechanism. But they sit in key positions. As you will see later in the talk with our translation program, we have actually hepatitis B vectors where we have packaged foreign cargos, and we're using the appropriate parts of that mechanism to make that possible. So even with the sparse number of packaging sites, it is actually possible to get that mechanism and the benefits of it. So let's look a little bit more into how that works. So here is work from Neil Ranson again, where you see cryo-EM density revealing one very clearly visible part of RNA, and this is showing basically the nucleation side of assembly. So this shows that there is commonality, and indeed the associated packaging signals are the ones that we're using for our translation work predominantly. But from a mechanistic point of view, let's look at what it means. So basically in solution, if you have just capsid protein, you have a population of particles with a T3, 180 protein, T4, 240 protein size capsid. But in the presence, again, of shorter fragments even, of the packaging sites, you are biasing this reaction towards the native T4. So it shows that geometry is being set by those packaging sites in some way. And that becomes important again for our application. So it switches the equilibrium. So before, the switch was between two dimers. Here, the switch is between different types of geometries and being mediated by different angles between the building blocks.
Can I just ask?
Yes, please.
So, but why would you then sort of see this
sort of only in one location mostly, right?
Good question, that's nucleation. So what I have glossed over a little bit, so there are different challenges that the virus faces in order to efficiently assemble. And the big barrier is nucleation. So this virus has the challenge of reverse transcribing later. So it wants to keep the nucleation, benefits for nucleation, without stitching too many other sites in that would potentially help with kinetic traps. But then it would also create a problem for reverse transcription. So it is especially the nucleation side. It's between 2 and 4 packaging sites, and we actually know it's 3 by now, but the density is consistent with 2 and 4. Yeah, good question. So I think it's always a trade-off between different challenges for the virus, and nucleation is one of the big ones. And nucleation theory is the big barrier. And also, if you think from the point of view of geometry, once you've set the geometry to a T4, it will not flip back to a T3, it will keep the curvature. So, and in this system, the Stockley lab have looked at antiviral therapy. So we worked with people at the NIH who had a system to scan for ligands that bind against RNA stem loops. So the RNA stem loops get immobilized. We submitted all the packaging sites we had identified, ligands were selected for, and then assembly experiments revealed which ones were working well. And indeed, there was in particular one compound that really bound with a good affinity and is a promising candidate. So this is basically a proof of principle that this is really a viable strategy as a clinical strategy. And these papers here are a mix of modeling and experiment where we basically show how this works. So this is just an upward that this really is a possible strategy, proven strategy. But I would like to.
So this is different than the other assembly inhibitors, which make often stable.
Different from the CAMs, because the CAMs do the geometry, but this one is actually this affecting the switch that sets the geometry, and not the other assembly inhibitor change the building block so that its curvature is different. And we just don't change the building block, we are changing the way they come together, the main difference. Yeah, they are actually quite good. We have a paper where we compare, so it's a theory paper where we compare these CAMs, I think they're called in hours, and we show that actually the best element from this list here is actually very competitive, is actually performing even better. I mean, most of them don't perform as good as theirs, but we have one that performs even better. So I can't probably be here with people interested in coronavirus. Now I can't give too much away on this, but I want to at least mention it because it belongs in this framework of viruses that we've been working on. So coronaviruses need not much introduction in this community here, but what we are interested in, as you can guess, is in how our mechanisms would potentially also apply here. And as you all know, the compaction step is already taken care of by nucleocapsid protein. The genome is basically binding around and is therefore a bit like in a chromatin-like organization compacted. But the question is there again some flavor of this mechanism that we've seen? So apart from the compaction, there is still this assembly and nucleation aspect that might be of interest. Now, in other coronaviruses, not SARS-CoV-2, it was known by Masters has a nice review here, but it's widely documented that there is a high affinity packaging signal in these systems, and it is basically conjectured that it works by bringing together N protein, M protein in the membrane, and the packaging side. It's not clear in which order these three components act together to nucleate assembly, but it is clear that they in some way do. So the question is there a way with our technologies to see if we can find something here? So now we had data, there's on the left is data, private communication from the Hamburg group, this is Kai Grunewald's group, where you can see these RNP complexes close to the membrane. You can see this also in this paper, the cell paper by Yao et al. So you clearly see there are these nice lineups of organization of these RNP complexes of the membrane. So the hypothesis arising from this is that perhaps you have these RNP complexes from this N protein anchored at M protein, and this sort of spanning the whole particle a little bit like a tent over this agglomerate in the middle. Now, that's why we started talking here, of course. You all know this much better than we do. So this is your work, Melanie, where you looked at the different tiles across the genome to see which package better. And clearly then, if this mechanism is relevant, it should actually reflect that and should be visible in these structures here. So now I have, because it's still work in progress, I have not put our signals or our proposed mechanism out, but we have used the same technology that I described earlier, knowledge of how many sites you have, what the geometric repeats are, to find potential candidates for the N and M binding signals. And the idea in a nutshell is, maybe explained best on this structure from the Yao et al., is that you have some anchoring up with M, and then you have these dimers of N protein that interact with the second type of site. So there are two types of sites here that interplay to do this. And so again, I drop a little bit over the sort of predicted sites, but we first, before we got in touch with you guys, validated this in leads. So seq1 is a sequence which is predicted to contain precisely the amount of signals to make one of those. And seq extended is the sequence that is predicted to make precisely two of those. So in leads, we had the opportunity to look at the genome with N protein only, but you can already see in the negative stain image quite nicely, we see more images on the next slide, that you get for the extended, these doublets, and the singlets for the shorter sequence. Also, if you mutate out the predicted N binding sites, you see agglomerates that you don't see nice assembly. So there is already like a sanity check for us that something is working as we hope it would. Now, taking the extended sequence then and looking at it in more detail, we wanted to see if it's really true that you can get these doublets. And yes, you can see it from Abid's work here, these doublets, so it works with the correct measures. The measures agree with the published measures for the singlets. So it appears that this is actually really, these units that we've identified might really be the nucleation sites for a singlet or doublet. And again, that is work in progress, but I don't want to go in too much detail. That's for a future talk, if it works. We'll have to see it work. Right, so now I want to segue. I hope oh yeah, no, that's good. I want to speak a bit more about the wider role in life science. I only spoke about assembly at the moment. And especially for the applications that you asked me about as well, assembly is not enough, it has to release. You can obviously batch it in, but if it doesn't come out, you have gained nothing. So we need to understand how this whole mechanism works. So I would like to go back to our favorite phage again, because that's where we can understand it best. And then we can transfer this to other viruses. So the next question for me now is the role of packaging sites confined to assembly, or is it actually a bigger system that we need to understand and potentially code into applications? Now, in order to do this, we have developed a new technique to interrogate packaged viral genomes. The reason is that chemical probing impacts on the structure of the virus. So we cannot therefore have a time series where we are tracking how packaging sites would change over time. But we can do this with this technique. So we've done it for the first time on viruses. Obviously, XRF synchrotron footprinting has been done on ribosomal proteins, so it's been around before, but together with the leads team, we are the first ones who've done it on viruses. So use this technique here. And that has thrown up a lot of technical problems for us because the typical analysis techniques did not work. So we needed to get a complete pipeline that we now have published, and that's available on GitHub and everybody can see it. So what's going on? So we basically submit the packaged viral genome, so the capsid is the genome, to synchrotron radiation. And what the radiation does, it excites the water and it changes ribose sugars if the portion of the RNA is single-stranded. So if there is a contact between RNA and protein, or if there's an RNA-RNA contact, it will be protected. But if it's single-stranded, it can potentially be modified. And then we can read this out. We can see where these nicks, where these changes are. We read this out, and this gives us then these plots where we have the x-axis being the genome, and then basically the peaks where we had the most modifications. So these peaks basically tell us where we had basically single-stranded portions. So very similar to chemical footprinting in this regard. So then we overcame a couple of computation challenges. Again, talk to me if you want to know. I just want to sort of come to more results if I can. So then, again, on the MS2 system, there is this known major packaging site and then neighboring packaging sites that were characterized before. As in chemical footprinting, you need to have some input to fix the parameters in the folding programs that tell you how to take into account those data. So these are typically what we call M and B values. This is a Boltzmann factor. It's an exponent M times data plus B, which is the offset. So in a nutshell, M tells me how strongly I want to consider those data for my prediction. And B is just making sure that I'm not going under a zero line, so it's a correction for dropping under the zero. So with this in mind, then what you do is you have these grids with M and B, and you look for which combinations known behavior is reproduced. You use then those combinations to make predictions all around. This is very standard. This is nothing new. Weeks would do the same with chemical footprinting. So you basically have these type of graphs, and that's the standards that we used. Now, why are we excited about this? Because we learned something we didn't expect. And it threw us off and again started a whole new pipeline for us, because we knew from Hong's data, Hongzhu has this quarium data where he characterized 15 packaging sites plus more stem loops with the characteristic features in the vicinity of the protein layer. But what we see here is actually that there is potential for transient PS, because apart from these 31 PSs we see actually in contact, we see 34 unbounded ones that are very close and could potentially at some point have also acted. So that has to do with the grading of the protection. So you see there's a certain probability for them to be in contact, certainly not in all particles in the ensemble, but clearly in some. So what we are concluding from this, therefore, is that potentially PSs, packaging signals, have acted during the assembly step, but then fell off or unbound, and the conformation of the genome has changed such that they don't reconnect. They stay dislocated. So what could this mean? And this is, I just learned shortly before the talk, as Greg Towers is also quite excited here. So basically, we are arguing here that there is a mechanism at play that uses this phenomenon. And this can be described with a phenomenon called molecular frustration. So in a nutshell, molecular frustration was introduced by Reidun to look at protein folding problems and looking at different conformations that are equally probable, but therefore have the possibility to have this allosteric control, because it's possible to go between these different conformations if they are of similar energy, of similar probability. So we say a system is frustrated if it admits very similar energy structures that could potentially coexist in an ensemble. Now, the way then we are arguing here is to look in the system MS2 again at the protein dimer, and we are plotting this so-called frustration profile. So frustration profile is as follows. You go along the amino acid sequence. You see this on the x-axis. And then you're asking in any particular position, so how many other possibilities do I have to reorient this into different contexts? So it's a measure for how much a certain amino acid is in a fixed position, or actually what kind of changes can, with equal energy or similar energy, can happen around. And what you see here, it is quite interesting, is you have basically a change in this pattern upon binding of the packaging site. So the packaging site is actually changing this frustration landscape. And so now knowing this and looking at the geometry of the capsid, so we profiled in which of those dimers in the capsid we have at a certain time t after it's been basically assembled for a while, in which positions we have packaging sites still present or where we lost packaging sites. So the red ones are the ones where we lost sites. The green ones is where we still have sites. And I haven't told you about the yellow protein. That's a single copy of a so-called maturation protein. This contacts the bacterial pillows for ejection of the genome. This is very important for the release mechanism. And what I want you to notice is that we have these accumulations of sites where we have lost the packaging signal around that maturation protein that we need to basically release first. So you can already guess what's happening. You destabilize the protein shell through the loss, the very coordinated loss of packaging sites. And that's coded by the affinities. So I haven't perhaps stressed enough that these packaging sites have different affinities, which is regulated both by their stability but by the variation around the core motif. So we know certain variations around the core motif have quite an impact on the affinities. And if you look at the sites that are sitting here, you see that they actually have this reduced affinity, so they are more likely to fall off. So basically from the onset, the phage has programmed in to release at that stage at a certain point. So we are arguing that.
Is the number of the sites also important? The
Yes,
number of the
absolutely. A number and position. And it's actually, it needs a certain number. You're absolutely right. It is not enough, it doesn't work. It needs enough of them to add up to an energy that's basically destabilizing. Absolutely. Yeah. Good point. And so basically what this then shows us is, so as a cartoon, so here's maturation protein again. These are the destabilized sites. It can actually easier pull out maturation protein, which then pulls out the genome. So maturation protein is bound to the genome and pulling the 3' end of the genome out into the host. Now, we were then wondering, because we always like to scale up, but first let's have a cartoon on this message again. So we have an assembly phase where we've discussed at length that packaging sites are important, but then there is this uncoating phase, which is also governed by that same mechanism. Now, we were then teaming up with Greg Towers, and we wanted to understand, is something similar happening in more complex viruses again? And here we are looking at different cofactors on the question as to, is there a sequential order in which they happen? Is there therefore a certain time arrow in an infection that is governed by this mechanism? So we have a joint paper here where we are looking at this process based with cofactors and how one cofactor influences the next one. So again, it's a phenomenon that happens across different viruses. I also have a collaboration here with Carol Teschke. This is again a big phage, so she's a phage person, but in phages, scaffolding proteins play similar roles to packaging sites. We don't really have an RNA genome here, but the question is, does some of that mechanism actually prevail? And it's not in the genome, but it is in these scaffolding proteins, and we're just writing a paper up. She's been as a Fulbright professor in York for 3 months, and we now understand how this works and how this is mediated by a similar mechanism as well. So it's a very broad mechanism, but obviously in different incarnations, so rather cofactors via scaffolding proteins in this situation. But I'd like to segue into our translation work. So the best way to do this is to answer the question that we did in our joint science paper with the Hilbert lab here, where the question is this actually a mechanism that is confined to viruses? Or is this actually much broader? Is this actually applicable to protein shells at large? So the Hilbert lab looks at bacterial enzymes that form dodecahedral shells, a bit like virus shells, from pentamers. And what they've done in this experiment, they have cyclically permuted the protein so that they could fuse the lambda binding site on one end that's pointing towards the interior, so that the messenger RNA, when flanked by the packaging signals of lambda, would be recognized by the system. So it's basically a bacterial enzyme that has been told how to recognize its messenger RNA, mediated by packaging signals from lambda. So what's really interesting there is they did different rounds of directed evolution in the laboratory. So they saw, first of all, that larger particle morphologies were assembling, which on its own is already quite interesting. But even more puzzling at that time was the fact that when the largest container morphology was achieved, there was another step of efficiency in packaging, a very drastic step in efficiency that could not be accounted with geometry alone. So the question is, can we understand what's going in here? So here is a picture of the different capsid geometries that become increasingly larger, up to this 240 protein structure here. It's a very interesting geometry because of the sort of had to choose for this talk a little bit. There's a nice domain swap, which I should probably also have talked about. We also have some modeling work that for the first time actually provides a mathematical framework to understand, classify these container geometries. So PNAS we had just coming out in December, but I thought you're probably more interested in the biological side of things. So I just advertised if there's someone who is interested in that. I'm here today. Ask me if there's anything you want to know. But I'd like to segue forward into what I think most of you probably want to hear from me, which is these data that show the packaging. So basically, you see these different rounds of evolution. At 3, the geometry doesn't change anymore. So from 3 to 4, this step in packaging of the cognate messenger RNA, the blue, is actually obviously not due to geometric change. So what we've done then here is we follow up this hypothesis that this is again due to evolution of this packaging mechanism. So we did our XRF technique here, looked at the structure, and what we could show is that indeed packaging sites similar to lambda have evolved and have made a cassette, which is quite interesting to us. So then at this point, Don Hilbert was rightly so very skeptical, and said, oh, maybe that's just obviously could have been a fluke in some way. Maybe these sites just arise by random chance. So he set us a challenge, and that's not yet published, but I keep it here because we are submitting it very soon. So he said then, well, I believe that you understand the mechanism if you go back to one geometric step below. So this is the 180 shell, the one step smaller. And he said, if you really understand it, you should be able to recode the genome for this one so that it is getting you better packaging. And here are the data. So that's what we're writing up right now, is here. So for some reason I haven't got time to go into, the C1 is most optimized with geometry as well, the knowledge of geometry. And you can see how much more efficient assembly is, both from those data, and you can also see it from this graph here, which clearly shows here's wild type. You see we've exceeded the efficiency of wild type massively here. So we can really engineer this, we can do this. And this is work that we're doing with the Stockley lab. Again, in response to your question, this is actually a foreign cargo now. So this is no longer a genome or a precursor genome. This is actually a completely different cargo with the hepatitis B delivery system. And basically coding this in we were able to make the assembly more efficient then even Walter as well. So we were quite happy when we got that. We're writing this up as well, as a couple of things in the pipeline. So
when you make the assembly more efficient, is the encoding also effective?
That is a very good question. At the moment, we have just looked at the packaging. You need data for the encoding. It's a good point. But we haven't coded too many in, so I have to hope, because with this system, that we haven't over-coded it. Yeah, but you're right, we have to check that question. Yeah. So what I hope I could show is that we get some mechanistic understanding from integrative systems virology, which really my trade is sort of bringing these different techniques together in ways to answer open questions. Hopefully I could show you that this has allowed us to develop novel antiviral strategies. We also work on viral vector design and gene therapy. We haven't worked much on vaccines yet, but that's in the pipeline as well. We can use that knowledge to make particles stable that otherwise would perhaps not be as stable as desired. Right, and I'd like to advertise, I'm not sure if there are any theoreticians, we are hiring. We got another big welcome round just now, so there in my team there are 2 positions and also experimental positions in the Stockley lab. So I have 2 positions. One would be going down the pipeline of understanding these packaging sites, identification of packaging sites, and the recoding work. So this is also partially with industry. Then this other work would be the mechanistic side, modeling mechanisms. I've probably spoken a little less about it or just touched upon it. There's more the mathematical geometric side as well. So if you're interested in either, application deadline is pretty much today, but sorry about that, but that can be bent a little bit. This can be bent. I always wait to bend it. And if anybody wants, I put this up here so you can scan the codes if there's anybody who's interested. Okay, and then finally, my thanks to my wonderful team. I mean, it's a pleasure. We're all having different expertise and bringing this to bear. And then also our team in Leeds, Peter Stock is the lead there, with the postdocs there, and many collaborators. I'm sure I missed a lot out. But yeah, so with this, thank you so much. And I'm around for the day and happy to answer questions on anything.